Dear students, welcome to the another video of Eduins. Today we will discuss about the monohybrid cross. What is monohybrid cross? From the name, that is mono mean one. That is the cross between two organisms with one pair of contrasting characters. Okay. The Mendel's monohybrid cross, of course, the cross was occurs between the pea plant. Two types of pea plant he was taken based on the which character, that is the height. Height of what? Height of stem. That is long and short. Okay. The long height pea plant or tall height pea plant was denoted as capital T and capital T as their genotype, and the short height is denoted as small t and small t. Okay, so in parental generation, that is P1 generation, the cross between capital T into small t, small t. This condition we know, this condition is known as homozygous condition, where two alleles are same, that is capital T and capital T. So the gamete will produce from here, that is capital T, and this is small t and small t. Okay, these are the gametes. Okay, so if in the F1 or filial generation 1, what kind of offspring will produce? Look, it will this one, this one, this one, and this one. So capital T cross with small t, that is capital T small t. The capital T with small t, that is also capital T small t. Here also capital T small t and capital T small t. So in F1 generation, all plants will be hybrid and tall. Why tall? Because we know that is tall height is the dominant character and short height is the recessive character. What is dominant and what is recessive? That is, the character will show its character in presence of only one allele. Okay, and in case of recessive, which will can only express when the two alleles are present. Okay, so in F1 generation, all plants are tall. Now he was done the cross between the F1 generation offsprings, like that is the cross between capital T small t and capital T small t. So this will be the P2 generation or parental 2 generation. This is the P1, these are the gametes, this is F1, and now this is P1. So what kinds of gamete will produce from this? One capital T and one small t. Here also one capital T and one small t. Now these are the gametes Now the results of the offsprings, their phenotype and genotype, will discuss by a table which is known as Punnett Square or checkerboard. Here we'll place the gametes. This is capital T. We write down the capital T here. This is small t. We write down the small t here. Now the capital T here and this is small t here. So this is the male. This is in case of female. Now this is very much easy to calculate. That is the capital T and capital T. So here will be the capital T, capital T. The capital T here and here the small t, that is capital T, small t. Now the small t with capital T, that is capital T, small t, and small t, that is small t, small t. So the offsprings will produce in three types. What are they? One is capital T, two capital T, small t, and one small t, small t. So what are the phenotype of these offsprings? This is of course capital T, tall. This is the capital T, small t. This is also tall because here present one capital T, and this is the short. So this will be the tall. This is also tall, and this is short. So what are the phenotype? Phenotype mean the external character. These two are same. That's why the phenotype one two tall and one short. That is the between them. This is tall 1, this is tall 2, this is tall 3. Okay. So the ratio will be 3:1. That is tall is to short. That is tall and this is short. Okay. Now the genotypic ratio. Genotypic ratio. Genotypic ratio, that is genetically they are same or different. Of course different, because look here, the capital T, that is homozygous dominant. Capital T is the homozygous dominant. Okay. Capital T, small t is heterozygous. Okay. And small t is homozygous recessive. Okay. So the genetic or genotypic ratio will be the homozygous dominant is 1, the heterozygous is 2, and the homozygous recessive is 1. So the genotypic ratio will be 1:2:1. Okay. So you have to remember this phenotypic and genotypic ratio and how the cross will happen. You have to write down in step by step, that is P1 generation, between which two parents we are crossing, then what types of gametes are produced, then what type of offsprings produced by F1 generation, then what is the genotype of the P2 generation parents, then their gametes, and lastly you have to do the Punnett square or checkerboard. Punnett square or checkerboard. Okay. Then you will get the conclusion and the phenotypic and the genotypic ratio. Okay. So that's all for the today's video. If you have any doubts, you may ask in our comment section. We will definitely discuss it in our next video. And if you like to get more such informative video, you may subscribe our YouTube channel Eduins, and also follow, comment and share our Facebook page Eduins. Thank you.
Eduins, learn, grow, succeed.